Hello, my name is Anora, and today we're going to cover some topics from the GIT Physiology. These are the topics that we're going to cover. So first we'll go through metabolism, then we'll talk a bit about calcium, about vitamins, then we'll go through food, the regulation of food intake, the functions of the stomach and what happens to food in stomach, and then what happens to food in intestines. So let's start with metabolism. We need to distinguish two different words, metabolism and energy metabolism. Metabolism itself is simply when enzyme takes certain molecules and convert them into other molecules through multi-step pathways. It has to do with how the body uses food to obtain energy and build molecules that it needs. Metabolic pathways can be of two varieties. These are catabolic pathways and anabolic pathways. Catabolic, where larger molecules are broken into smaller ones, and anabolic pathways are the other way around. When we build up molecules, consuming energy. Energy metabolism is actually the set of chemical processes to extract energy from food. So it's a bit different from metabolism itself, all right? So we extract energy from food and we use it in the body. The energy we obtain is chemical energy, and chemical energy is the energy in a substance that can be released by a chemical reaction. So just let's imagine this is our food that we consume. In our body we change it to some substances, and out of those substances we get energy. Let's take a look how it happens. The energy is measured in calories, and we have 4 sources of energy, and out of all sources we can say how many calories we can get out of 1 gram, for example. The 3 of them are shown here, and the 4th actually is alcohol, but we're not going to cover it today. Actually, there are other substances that we consume, such as water, vitamins, minerals, but we do not have the chemical processes to get energy of them. The energy, and this is very important part, all right? The energy in these foods, proteins, carbs, fats, and actually in alcohol as well, ethanol, is in carbon-hydrogen bonds. Carbon-hydrogen bonds, and this is what you need to keep in mind. The more CH bonds a substance has, the more energy we'll get out of it. That's why fats have 9, whereas carbs and proteins 4 calories per gram. Let's draw a bit. Whatever the energy yielding nutrient is, they all go through 3 basic processes. 1, 2, 3. First is conversion to acetyl-CoA. And we need to keep in mind the number of carbon molecules. In acetyl-CoA, we've got 2 carbons. This is considered the center of energy metabolism. Once we have this 2 carbon molecule, we attach it to 4 carbon molecule, and we get 6 carbon molecule. And this 6 carbon molecule, we're going to send through the TCA cycle. In TCA cycle, we're going to take 2 let's draw it with this. We're going to take 2 of those carbons, add oxygen to it, and we're going to get carbon dioxide. Then 4 carbon molecule that is left meets again acetyl-CoA, which is 2 carbon molecule, and we regenerate the cycle. If we think about the carbon-hydrogen bonds that we split apart to get the energy, we know what happens to the carbons, but we still have 2 hydrogens left, right? These are actually thrown off in the system via 2 carrier molecules, which are NAD and FAD. These carrier molecules pick up these energized hydrogen electrons and enter so-called electron transport chain, which is the 3rd basic process that we need to know. In response to all these processes, the body will have 3 end products, which are carbon dioxide, water, and a bunch of ATP. This is the molecule we need in order to get and use energy. Now, let's take a look at each energy-yielding nutrients and how they get through these 3 basic pathways. Let's start with carbs. Mainly carbs are converted into glucose, but please keep in mind, right, that all I'm talking about is very, very shortened. It doesn't contain very more complex ways. It has like carbs are not consisting of glucose only, right? We have other sugars as well, but we're gonna keep it as simple as possible, right? If you want to recall some biochemistry processes, please go to biochemistry books. So let's continue. Carbs are like we mainly get from carbs glucose, right? And glucose is 6 carbon molecule, which is converted afterwards into pyruvate in the process called glycolysis. This process up to here doesn't require oxygen. That's why it's called anaerobic. But afterwards, we need oxygen in order our pyruvate, which is 3 carbon molecule, to convert it to acetyl-CoA. Pyruvate is 3 carbon molecule. Acetyl-CoA is 2 carbon molecule. It means that we take 1 carbon molecule, add oxygen to it, get CO2, and we're left with acetyl-CoA, which is the first basic process that we need to come to. Let's go to proteins. Proteins consist of amino acids. So what do we get from amino acids? First, like amino acids, it means that they contain not only CH bonds that we need, but also nitrogen. And what happens to nitrogen? Nitrogen actually is clipped off out of amino acids by the process called deamination. And which is converted to urea, and urea goes to urine, and we get rid out of it. And what we're left with is a carbohydrate base that we need. that has those CH bonds, right? There are 20 amino acids, different amino acids, and each of them has its own pathway. Some of them convert directly to pyruvate. Some of them go to acetyl-CoA, are converted into acetyl-CoA, and some of them go directly to the TCA cycle. The amount of amino acids that are converted into pyruvate is approximately 50% out of that one. The other half is over here. Let's go to fats. I have written here triglycerides. These are actually a type of fat found in blood, right? And what do they consist of? They consist of glycerol as a backbone, and which has three carbon molecule, and three fatty acids, right? These triglycerides are split into glycerol and fatty acids separately, and each of them has its own pathway. Glycerol, being 3 carbon molecule, is converted directly to pyruvate, while fatty acids are converted into several, actually many acetyl-CoAs via the process called beta oxidation. This is what I wanted to draw right now, and just in case you want to print screen it, this is a better version of what I was trying to show here. Let's go to the measurement. Regarding measurement, metabolism is measured by determining how much oxygen your body consumes over a specific amount of time. There are two rates that we're going to cover, the BMR and RMR. BMR, or basal metabolic rate, is a minimum energy required to sustain life. This energy is needed to sustain basic body functions at rest, such as breathing, circulation, kidney function, etc. So that's why it is measured while the person is awake, but at complete rest. So it is often conducted in a darkened room upon a person's waking up or after at least 8 hours of sleep. The second rate we're going to talk about is RMR, which is resting metabolic rate, and it is actually BMR plus calories needed for basic daily activities. But it doesn't include, please keep in mind that it doesn't include calories for heavy work or exercise, right? Calorie counters and calculators usually use resting metabolic rate rather than the basal metabolic rate, because actually the conditions upon which the RMR rates are taken reflect the normal situation in a person's day-to-day activities, right? So the results are more realistic. RMR is commonly measured in clinical settings using indirect calorimetry, as I've said before, which measures the amount of oxygen consumption and carbon dioxide production, as shown here, over here. But actually we can measure it through heat production, but it is not widely used. Now we're going to talk a bit about calcium. Calcium is an essential ion that is needed for a bunch of physiological processes in our body. When you're hearing calcium, first things first, what you're thinking about are bones and teeth. So bones and teeth contain calcium as an essential building block. What else does calcium do? It actually has much more wide roles in our body. It actually enables muscle contraction. Not only in skeletal muscles, all right? So if we take skeletal muscles, in skeletal muscles, calcium is taken up from both extracellular space and intracellular. Within the cell, we store calcium in sarcoplasmic reticulum. Why it is dangerous for us to have high levels of calcium ions in our cells freely? Because calcium also is responsible for activating different enzymes, hormones, and other molecules within the cell. So if we have a lot of calcium within the cell, it means that it can activate different enzymes and molecule substances that we do not want to activate at a certain time. That's why within the cell, calcium is stored in the sarcoplasmic reticulum. Please keep it in mind. I'm not the best painter, but I hope you understand. Also, it is very important for the normal functioning of the heart. It is actually responsible for its contraction, as I've told you, muscle contraction, right? And in order for myocardium to release calcium ions from the sarcoplasmic reticulum, it requires extracellular calcium. So it can be said that myocardial contraction depends on plasma calcium concentration, while skeletal muscles use both extracellular and intracellular stores. Calcium actually also is an essential. So here we need to draw heart as well. All right, it is very important for heart. Let's go further. Calcium is an essential part of a blood clotting cascade, coagulation cascade. As I've told you before, it actually activates a large number of enzymes, and it serves as second messenger in the cytosol of cells. A second messenger means that it mediates the effects of some hormones that have receptors on the membrane. So it is very important to regulate anything, almost anything in our body. Also, it regulates neuronal excitability. Increased concentration extracellularly stabilizes the membranes and reduce cell excitability or irritability, while increasing intracellular calcium increases cell irritability, as I've told you before, because it is a second messenger. When the concentration of ionized calcium in the plasma changes, the excitability of most cells changes in the first place. This is what you need to keep in mind. In our body, almost 75% of calcium is stored in bones. If we take calcium in plasma, the normal plasma calcium concentration is 2 to 2.75 millimoles per liter. Of this, 50% is ionized. It means that it is free. We've got free calcium, 50% of it. 40 to 45% of it is bound to proteins, and the rest is forming complex calcium, like calcium bicarbonate, calcium phosphate, calcium citrate, etc. Ionized calcium is mainly important for myocardial and neuromuscular excitability. So let's draw it here for you to remember it. If we talk about the ratio of bound calcium to ionized calcium, it depends on the amount of protein in the plasma. So let's think about it like that. Our proteins bind calcium. If we have increased concentration of plasma proteins, we've got increased capacity of calcium binding. So it means that calcium is more bound to proteins rather than ionized and free, so it leads to decrease in ionized calcium. Also, pH in the blood has a huge role in it. Let's think about it like that. Proteins are working as a buffer, which means that they take up the hydrogen ions when needed. So, but in order to do so, they need to increase their capacity to bind hydrogens. And in order to do so, they need to release calcium back to plasma. So let's think about it like that. If we have acidosis, which means that we have lower pH in blood, it means that we've got a lot of H pluses. We've got a lot of H plus ions, which means that proteins need to do their role as buffers, so they need to bind those H plus. In order to do so, they release calcium and bind protons. It leads to increase in calcium ions, like in ionized calcium in the plasma. If we have alkalosis, which means that we have low amount of H plus in our blood, it means that proteins need to release this H plus back to blood plasma, and thus they have higher binding capacity for calcium, so it leads to decrease in calcium levels, ionized free calcium levels. The regulation of calcium is mainly regulated by 3 substances. Calcitriol, which is not shown here, but I will write it down here. Which is actually vitamin D, the active form of vitamin D, calcitonin, and PTH. What you need to remember is that calcitriol and PTH increase blood calcium levels, whereas only calcitonin lowers its levels. Let's take a look. What do they do? If we take the PTH, parathyroid hormone, it actually goes to kidneys and increases the calcium reabsorption. It goes to small intestine, it increases the calcium absorption, and it goes to bones, and it increases bones resorption. So it's not good for bones. It means that we get a lot of calcium. If we take calcitonin, It goes again to kidneys, but it does the other way around. It lowers calcium reabsorption in kidneys. It lowers calcium absorption in small intestine. It lowers bone resorption. So it means that we've got lower blood calcium. Calcitriol goes to all of them as well. And for example, in small intestine, they increase calcium absorption. In kidneys, they increase calcium reabsorption. But in bones, they do not increase bone resorption. They actually put calcium into the bones. And we're going to talk about vitamin D in more details in a bit, because we're going to go through vitamins. Vitamins is the topic that is actually studied every year of medical school, like starting from the first year of your medical school till the 6th. So let's go through them. We've got two groups of vitamins, fat-soluble and water-soluble. If we're talking about fat-soluble vitamins, it means that they are hydrophobic. So it means that in order to get absorbed, they need lipids. And while they need lipids, they need bile salts. Water-soluble vitamins do not need either of them. So they have simple intestinal absorption, and actually they use just specific luminal transporters, but it's much easier than in fat-soluble vitamins. Regarding transportation in blood, again, if we're talking about fat-soluble vitamins, they are fat-soluble, they are hydrophobic, so they need transporter, like carrier protein. While water-soluble, they do not, except vitamin B12. Storage. Again, fat-solubles are fat-soluble vitamins. That it means that they can get stored in liver and fatty tissues, and it is very unlikely for them to get excreted because they're fat-soluble. Whereas water-soluble vitamins, due to their hydrophilic nature, they are not stored because they got excreted by kidneys. And regarding accumulation, when we accumulate, in fat-soluble vitamins, we can see hypervitaminosis, and we'll go through those symptoms. Whereas in water-soluble vitamins, it is very unlikely for them to have hypervitaminosis. When we have deficiency, which means that we have low amount of fat-soluble or water-soluble vitamins, in fat-soluble vitamins, deficiencies are caused usually by malnutrition or steatorrhea, called by malabsorption, or bile acid deficiencies, or by different medications or supplements, or some different genetic disorders. While in water-soluble, deficiencies are usually caused by restricted diet, but they do not require any bile salts or anything like that. That's why they do not have, we do not see deficiencies of water-soluble vitamins when we have bile acid deficiency. But for example, malabsorption disorder such as gastritis or gastric resection or anything like that, and congenital disorders. It is important for you to understand that water-soluble vitamins are B complex vitamins and vitamin C, and deficiency of B complex can cause common symptoms, which are glossitis, dermatitis, and diarrhea. You need to keep that in mind. We'll go through it again in a bit. Let's go through fat-soluble vitamins first. You need to know all of them, that these are vitamin A, D, E, and K. Vitamin A. What do you think about when you're thinking about when you hear vitamin A? First, vision, as well as vitamin A actually affects expression of certain genes in cell nucleus. It is an antioxidant, and very importantly, it promotes cell differentiation and specialization, and we're going to talk about it, why it is so important. If we take the deficiency of vitamin A, it is characterized by a malfunctioning synthesis of glycoproteins and glycolipids. Why so? Because these are the substances that are synthesized by the use of vitamin A. That's why we have the cell differentiation and specialization, thanks to those glycoproteins and glycolipids, the synthesis. So if we have low vitamin A amounts in our body, it leads to malfunctioning synthesis of glycoproteins and glycolipids, and by not differentiating and specializing cells, we can get immunosuppression, right? As well as the differentiation and specialization is very important for our epithelial cells and our skin. So when we have deficiency in vitamin A, the typical resulting symptom is keratinization of epithelial tissues, mucosal atrophy, and reduced secretion of mucous glands. If we're talking about hypervitaminosis of vitamin A, it can lead to a variety of symptoms like liver damage, nausea, vomiting, diarrhea, weight loss, alopecia, increased bone fragility, even, and bleeding. But very significant is its teratogenic effect. It is very, very important. Because again, it promotes cell differentiation and specialization. That's why it has teratogenicity. Okay, let's go through vitamin D. Vitamin D is, as we've already talked about, it is very important in the regulation of calcium and phosphate metabolism. Its active forms, like calcitriol, stimulates synthesis of proteins involved in the absorption of calcium and phosphate in small intestine, as I've told you before, and thus it ensures their availability for bone formation and collagen synthesis. So we can think about what happens if we do not have enough amounts of vitamin D in our body. We've got hypercalcemic tetany. We've got osteomalacia in adults, and if our patient is a child, it has rickets. I guess you've heard of it for sure. The toxicity leads to hyperglycemia, loss of appetite, and different kidney diseases, because it can lead to calcium salt deposition in various tissues, kidneys also. If we talk about vitamin E, what you need to know about vitamin E is that it is antioxidant, right? This is very important. It is one of the main antioxidants. If we talk about vitamin K, very important its regulation, its role in coagulation. So if we do not have vitamin K enough, we have coagulopathy. If we have excessive vitamin K, it can lead to hemolytic anemia, and all the consequences of it. Let's go to water-soluble vitamins. Let's start with general. In general, well, water-soluble vitamins are B complex and vitamin C. And if we're talking about B vitamins, these are used in many different metabolic pathways. Its the B vitamins complex deficiencies have greatest effect on rapidly growing tissues. There are actually several common symptoms, as I've told you before, and you can, if you think about almost any vitamin B deficiency, you can put there these symptoms. These are dermatitis, glossitis, diarrhea, and cheilitis. Dermatitis is a rash on the skin. Glossitis is swelling or redness of tongue. Diarrhea is diarrhea. And cheilitis is skin breakdown in the corner of the lips. We're not going to go through all of these vitamins. We'll go through only some of them. Very important, vitamin B1. It is a coenzyme for various reactions involving dehydrogenase enzymes and carbohydrate and amino acid metabolism. So we've got like a bunch of reactions that require that vitamin B1. So if we develop deficiency in vitamin B1, we develop a disease called beriberi. Beriberi is characterized by an impaired saccharide and amino acid metabolism, and its symptoms include peripheral myopathies, fatigue, anorexia. Then it can even lead to edema and different other disorders. Also, very important for you to know is that so-called Wernicke-Korsakoff syndrome is a very kind of common disorder of chronic alcoholics. Mainly, it is developed as neurological condition, and it's like a neurological condition. So those patients develop many neurological symptoms. But what you need to know is that when you have a patient that is chronic alcoholic and he has Wernicke-Korsakoff syndrome, all you need to do is to give him vitamin B1 as well as glucose, because vitamin B1 is involved in metabolism of carbs. So it means that he's hypoglycemic at the time. So you need really to think about both of these things. So when you see Wernicke-Korsakoff syndrome, you give him both B1 and glucose, and by that you can remember that B1 is involved in carbs metabolism. Vitamin B2, riboflavin, is we've already actually mentioned its active form which is FAD. It actually can be in the form of FMN, like flavin mononucleotide, but it is less common. So thinking about what does FAD, you can think about what we need B2 for. If we're talking about B3 and niacin, before it was known as vitamin PP, from the word pellagra, preventive, and what we need it for NAD and NADP. molecules. And you recall that NAD molecule is very important in our electron transport chain. NADP molecule is used in ribose metabolism. So both of them are very important and widely used in all cells. So if we have deficiency in vitamin B3, this deficiency, patient develops a disorder called pellagra, and you need to know that pellagra is disorder of 3D, like 3D disease, which are dermatitis, diarrhea, and dementia. Vitamin B5 is not that high yield, so but you need to know that it is a vital component of coenzyme A. B6, again, it participates in the metabolism of amino acids like transaminase or decarboxylase. Vitamin B6 deficiencies quite rare, and as you can see, usually have those common symptoms in the deficiency. If we take vitamin B7, biotin, again, it is not that much high yield, so deficiency is rare, and it's mainly a factor for carboxylases, transcarboxylases, and decarboxylases. Let's go to vitamin B9 and B12. We're going to cover it together because these are very high yield vitamins. If we're talking about vitamin B9, so it is called folate, and vitamin B9 is actually, both vitamin B9 and vitamin B12 are used in the synthesis of DNA bases, DNA RNA bases. And both of them are the clinical manifestation of both B9 and B12 is more or less the same. But what you need to know is that in vitamin B9 deficiency, you do not have any neurological symptoms, whereas in vitamin B12, you have neurological symptoms. And this is very high yield. So if you have a patient that has microcytic megaloblastic anemia, which is caused either by B9 deficiency or B12 deficiency, you need to look at him and think whether he has or not any neurological symptoms. Because if you give a patient with B12 deficiency vitamin B9, the anemia or other symptoms will be, you'll get rid of them. But neurological symptoms will remain, and it can lead to very, very bad consequences. If we're talking about vitamin C is an antioxidant, and it is actually plays a huge role in collagen synthesis, and it facilitates iron absorption. So if you think about it, first thing that comes to your mind is when you have a vitamin C deficiency, you have scurvy. When you have toxicity, like when you have hypervitaminosis of vitamin C, you've got a patient can have fatigue, nausea, vomiting, diarrhea. If a patient is predisposed, he can have high risk of iron toxicity. Now let's go to food intake regulations. Food intake is affected by many factors. It is controlled from the hypothalamus, which we can see shown here. Let me take other pen color. This is the hypothalamus, right? So in the hypothalamus, we have a nucleus called arcuate nucleus, which plays a huge role major role in food intake regulation. At every moment, hypothalamus processes large amount of signals, which can actually be divided into two separate categories. Let's write it down here. Orexigenic and anorexigenic. If we take orexigenic, it means that these stimulates are appetite stimulating. If we take anorexigenic stimulates, these are appetite inhibiting. So, therefore, there are two sets of neurons with opposing functions in the arcuate nucleus. The first one is appetite stimulating, this one, and the second one, the right one, is appetite suppressing, and it produces POMC, and the orexigenic part of ARC is produces two main factors, which are NPY and AGRP. These substances are just like the substances that are orexigenic or anorexigenic. Neurons of this nucleus, arcuate nucleus, project to other nuclei of hypothalamus, of which the PVN or paraventricular nucleus is the most important. PVN neurons further process the information and project to other circuits outside the hypothalamus. And this is how hypothalamus coordinates the response that control energy intake and expenditure. So it goes further. These centers are actually affected by the whole set of information. For example, impulses from cortex, reflexes, conditioned reflexes, right? So, such as looking at food, talking about food, or looking at a person who eats. Genetics. Genetics actually affect on certain metabolic pathways. For example, there is a group of obese patients who do not produce heat during food processing and store all energy as a storage, as a fat storage. The temperature of the blood flowing through the hypothalamus can lead to changes in its work, in ARC work. Warm blood actually reduces food intake. That's why in high heat person has decreased appetite. Activity of glucostatic cells in the hypothalamus itself, which these cells are actually taking the amount of glycemia, and if we have lower glucose in our blood, or if we have higher glucose, like the amount of glucose leads to activation or deactivation of a food intake center. Stress. Actually, in moderate stress, food consumption increases. And on the contrary, in high stress or depression, it decreases. Some pro-inflammatory interleukins. That's why when we have fever, when we catch cold and we have fever, we have reduced appetite. So interleukins, I would say. Pro-inflammatory, they decrease the appetite. All right. And the last but not the least, some hormones. There are a bunch of hormones. Let's say thyroid hormones, cholecystokinin, a glucagon-like peptide 1, bombazine, etc., etc. And we're going to go through some of them. In order to go through some of them, let's divide our regulation of food intake into short-term and long-term regulation. I need to erase. All right. So in order to first, we're going to talk about short-term. Okay. In short-term regulation, there are several hormones that play a huge role. First is cholecystokinin, CCK. It is produced by duodenal and iliac cells, and the stimuli for its secretion are luminal stretch and biochemical changes linked to food passage. Actually, why short-term regulations? Because they depend on whether our stretch receptors in our GIT tract are activated or not. So if they are activated, we've got CCK, and it has the potential to cause the satiety, so it decreases our appetite. CCK actually goes to, let's draw it like that. CCK will come here and stimulate this part of the arcuate nucleus, which lowers the appetite. This increases it. Then we've got ghrelin. It is a hormone produced from the stomach about 2 hours after it's emptying, and actually it increases hunger. So ghrelin would be here. If we take GLP-1 or glucagon-like peptide 1, it is also a satiety signal. So we'll put it here, GLP-1. It acts as peripheral anorexigen in the same manner as CCK. So I'll just add it over here. But besides this role, it also stimulates insulin release from pancreas by potentiating glucose effect on Langerhans islets. If we talk about long term regulation, it actually depends on 2 hormones, leptin and insulin. And why long term? Because it depends on the amount of fat we have. If we have a lot of fat, it means that we need to eat less and move more. So in this manner, a hormone produced by adipose tissue called leptin works. It actually reduces the feeling of hunger, it increases basal metabolism, and it induces willing to move. So leptin would go here. Sorry. Would go here. If we have insulin, exhibit strong anorexigenic effect. We know that insulin is released after food intake, so it makes sense. Now let's talk a bit about stomach. We can think of a stomach as some sort of a muscular tube, right? It receives food from esophagus. It stores food for 1 to 3 hours. It mixes it with gastric secretions until it forms chyme. I'll write it down here. Chyme. And mechanically processes it. Afterwards, it slowly empties to the small intestine at a rate suitable for proper digestion and absorption. So it is a very smart organ, I would say. Let's take a look at its regions, all right? So stomach has cardia, which is an entrance of stomach. It has fundus, body, antrum, and pylorus. When food enters the stomach, a vagovagal reflex from stomach to brainstem and back reduces the tone of muscular wall, and the relaxed stomach is able to accommodate about 1.5 liters of food. Mixing of food with digestive juices is performed by mixing waves, moving towards the antrum. The waves are initiated by the basic electrical rhythms and consist of slow waves. So first we have slow waves. When progressing the waves from the body to antrum, they become more intense, providing very powerful peristaltic constricting rings that force the antral content towards the pylorus. So by increasing these waves, by making them more intense, it can lead to stomach to empty itself to the duodenum that we have afterwards. Actually, peristaltic waves provide a pumping action called pyloric pump. In the pylorus, here, the thickness of the circular muscle is greater than in earlier portions of the antrum, and thus it remains tonically contracted. This side is called pyloric sphincter. It prevents passage of food particles until they are completely mixed in the chyme to a fluid consistency. So it takes some time, 1 to 3 hours. Then, as I've told you before, the food goes to duodenum. There are 2 groups of factors that regulate the rate at which stomach empties to the duodenum. These are gastric factors and duodenal factors. And gastric factors actually, like the increased food volume in the stomach promotes its emptying, which is very logical. So if we think about the stomach wall, when it stretches, it elicits local myenteric reflexes that excite the pyloric pump and decrease the activity of pyloric sphincter. This gastrin, which is a hormone produced by the antral mucosa, here, has a mild activation effect on motor function of the stomach as well. Regarding duodenal factors, these are usually inhibiting. They inhibit the stomach emptying, and these factors are low pH, because we do not have the lining in the duodenum that prevents us from low pH eating up our wall. So low pH is bad for duodenum, so it's inhibiting factor. Presence of fat in the chyme as well. So overall, emptying of the stomach is controlled only to a moderate degree by stomach factors, such as filling, excitatory effect of gastrin, and the more important role of the control of stomach emptying has duodenum. These two mechanisms work together to slow the rate of emptying. When, for example, too much chyme is already in the small intestine, right? So it inhibits, right? Or if the chyme is excessively acid, as I've told you before, right? It is bad for duodenum. Or it contains too much unprocessed protein or fat, or it is hypotonic or hypertonic, or it is irritating. So anything that is bad for duodenum inhibits the stomach emptying. If we talk about gastric secretion, the body of stomach, this part, has contains glands that are called oxyntic or parietal glands. These, here they are. I'll take another pen. Here they are, parietal or oxyntic glands, and they secrete HCl mainly, as well as pepsinogen, intrinsic factor, and mucus. If we're talking about the antrum of stomach, let's take a look. Where is antrum? Antrum is here. It contains pyloric glands, which secrete mucus, as you can see here, to protect pyloric mucosa, right? Gastrin and some pepsinogen. Gastrin, we've already talked about it, but we'll talk about it even more. If we talk about HCl secretion, which is in the parietal cell of oxyntic glands, its secretion is kind of complicated. So it has several steps, so let's go through them. CO2 formed during some metabolism combines under the influence of carbonic anhydrase with water, and it forms bicarbonate and H+ ions. Sodium ions are actively transported out of the lumen as a response. K+ ions, potassium ions diffuse into the canaliculus, right? So So we have potassium ions in the canalicus, in the lumen of gastric pit. If we take those H+ ions, hydrogen ions, these are actively exchanged for these potassium ions in the lumen through the action of the proton pump. So in this way, the potassium is effectively recycled. On the other hand, bicarbonate is transported out of the basolateral membrane through here in exchange for chlorides. The outflow of bicarbonate into blood results in a slight elevation of blood pH, and it is actually known as alkaline tide. I'll write it down. So we have chloride ions and a bunch of hydrogen ions. If we talk about gastric secretions in general, first let's see which substances are produced in the stomach. We've already talked about the gastric glands, which are pyloric and oxyntic glands. Let's take a look at each substances. So we have mucus. Mucus is very important in our stomach because it is a physical barrier between lumen and epithelium. Because in the lumen we have very low pH, which is very irritating, would be very irritating for any mucosa. Bicarbonate. Actually, bicarbonate buffers gastric acids to prevent damage to the epithelium. And we have a lot of bicarbs in duodenum. Then we have gastric acid, HCl, which activates pepsin and kills bacteria. That's why it's very important. Pepsin, we actually have pepsinogen, and then it is activated by HCl to pepsin, and pepsin digests proteins, and it is very important. If we do not have well digested proteins in our duodenum, in our chyme, it can inhibit the emptying of stomach, as we've talked before. Then we have intrinsic factor. Intrinsic factor is very important for B12, vitamin B12 to absorb. Vitamin B12 actually combines with intrinsic factor in stomach and goes to ileum and is absorbed there. As I've told you before, the vitamin B12 is very important for DNA synthesis, and if we have the vitamin B12 deficiency, it can lead to megaloblastic anemia with neurological conditions. As well as the gastric secretion contain histamine. Histamine, as well as gastrin, and somatostatin, they stimulate or inhibit, they regulate gastric secretions. You need to know that somatostatin inhibits gastric secretions, gastric acid secretions, and actually it inhibits many other things in our GIT. And somatostatin, for example, is given to patients that are after different operations on their stomach or intestines. We've got gastric lipase, which digests fats, and gastrin, which again stimulates gastric acid secretion, as well as it has a small minor role in the emptying of stomach, as we've talked before. If we talk about the regulation of gastric secretion, we need to think about it as a huge system. The nervous system and the endocrine system collaborate in the digestive system to control gastric secretions. Not only secretions actually, but also motility associated with the movement of food throughout the gastrointestinal tract. Gastric activity involved in digestion is divided into 3 stages, and we need to know these stages because these stages show us how the gastric secretions and motility are regulated, not only gastric but gastrointestinal in general. The first phase is called cephalic phase. By the way, these phases overlap, and they all 3 can occur simultaneously, right? So just do not think that one starts, ends, and the other starts. So like they can overlap. The first phase is cephalic phase or cephalic stage. It occurs before the food enters the stomach. The cephalic phase results from smell, sight, thought of a food. So we've got neurogenic signals that cause cephalic phase originate in the cerebral cortex and appetite centers, which are amygdala, hypothalamus. And actually, this phase of secretion normally accounts for about 20% of the gastric secretion associated with eating a meal. But again, if a person doesn't like the food he sees, he won't have this phase, alright? So it's only like the cortex is working. That's why it's called cephalic. The second phase is called gastric phase. It accounts for about 2/3 of gastric secretions. Ingested food stimulates gastric activity by stretching the stomach and raising the pH of its contents. And this is not what stomach is used to, right? So this causes a cascade of events that actually lead to the release of HCl by parietal cells that lower the pH back and break apart the food. And actually, gastric secretion is stimulated chiefly by 3 chemicals. These are acetylcholine, histamine, and gastrin. If we have acetylcholine, it is secreted by the parasympathetic nerve fibers of both the short and long reflex pathways. If you can see, we've got long vasovagal reflex and we've got short reflex. Both of them release ACH. It actually excites secretion of all secretory cell types. So it enhances the secretion of pepsinogen, HCl, mucus. Actually, it plays a huge role in the first part as well. Cholinergic nerves signals originating in the vagus pass to the enteric nervous system of the stomach wall, and to the gastric glands. So cholinergic signals activating gastrins act via gastrin-releasing peptide called bombazine. So we can say that gastrin is stimulated by ACH. Through bombazine. But actually, gastrin secretion is also stimulated by peptides and amino acids in the stomach. Then we've got histamine. It is a paracrine secretion from the enteroendocrine cells in the gastric glands, and its main function is to excite the secretion of HCl. If we're talking about gastrin, we've already actually talked about it. It is a hormone produced by G cells in pyloric glands, and it takes part again in activating the HCl production, as well as it takes a small part in the emptying process. All three of these stimulate parietal cells to secrete hydrochloric acid and intrinsic factor, right? So all of them are stimulants. If we go to intestinal phase, which is the third and the last phase, why intestinal? Because we are emptying our stomach to the duodenum. So the stretching of the duodenum enhances gastric function by the vagal nerve, as the chyme causes the secretion of gastrin, which stimulates the stomach. So we trigger here enterogastric reflex, which is actually when the duodenum sends inhibitory signals to the stomach by way of enteric nervous system, all right? And enteroendocrine cells stimulate, like the chyme stimulates enteroendocrine cells of the intestine, and these release compounds that stimulate the pancreas and gallbladder. So if we talk about pancreas, it is stimulated by these cells by this phase, while also actually it suppresses gastric secretion and motility. How? By inhibitory hormones such as gastric inhibitory peptide, vasoactive intestinal polypeptide, and somatostatin, which we've already talked about. And we've talked about CCK, cholecystokinin, which plays a stimulating role for pancreas and bile duct. Let's go to pancreas. First, let's take a look at its basic functional morphology. So this is the pancreas, right? This is it, which opens up to duodenum through its ducts. So it has ducts and it has parenchyma. And parenchyma actually consists of pancreatic islets, which produce insulin and glucagon, but it is not what we need now. As well as this parenchyma has acinar cells that secrete digestive enzymes, not only digestive enzymes. As well as we have pancreatic duct, and it is also important in the secretion of pancreatic juice, because the epithelial lining of pancreatic ducts enables the production of and secretion of bicarb. Why do we need bicarbs in the duodenum? Because chyme entering the duodenum has very low pH, and duodenum doesn't have enough mucus as does the stomach in order to protect itself. That's why it needs to neutralize its acidity by getting bicarbs from the pancreas. So what does pancreas actually? By producing its juice, it contributes to the main part of absorption, of processing of the chyme of the food that enters the intestines. Let's take a look at it. What does it do? Pancreatic juice contains water and solids, and we need to know which substances it has. It has actually a lot of substances, but we'll take a look at digestive enzymes and bicarbonate. If we talk about enzymes, take a look. It has many proteolytic enzymes. It has lipolytic enzymes, a lot of them, and amylolytic enzymes. Do you know what is amylolytic enzymes? These are pancreatic amylase, and these hydrolase starch, glycogen, and other carbohydrates. If we're talking about proteolytic enzymes, this is trypsinogen, right? Like the pancreatic juice is trypsinogen, hemotrypsinogen. What does it mean? That it means that these enzymes are not in an active form. They are in inactive form. Why? In order for pancreatic juice, in order for these enzymes not to digest the pancreas itself. So trypsin, we get trypsin, hemotrypsin, and other enzymes activated in the duodenum itself. So before they enter the duodenum, before they enter the intestines, they are not active, all right? If they were, the pancreas would eat itself, digest itself. Just take a look at all these enzymes and just think about it. We need to digest everything. We need to digest both protons, lipids, and carbs. We need to digest everything. So pancreatic juice has it all, I would say. As well as I've said, pancreatic juice has a large amount of bicarbonate in order to neutralize the pH of the chyme that enters the duodenum. If we talk about the regulation, as you might accept, secretion from the exocrine pancreas is regulated by both neural and endocrine controls. We've already actually talked a bit about it. During interdigestive periods, very little secretion takes place, right? Because it wouldn't make sense. But as food enters the stomach, and a little later, chyme flows into the small intestine, pancreatic secretion is strongly stimulated. Like the stomach, the pancreas is innervated by the vagus nerve, which applies a low-level stimulus to secretion in response to anticipation of a meal. However, the most important stimuli for pancreatic secretion comes from 3 hormones secreted by the enteric endocrine system. If you recall, in the intestinal phase, these are secreted. First is CCK, we've already talked about it, cholecystokinin. This hormone is synthesized and secreted by enteric endocrine cells located in the duodenum here. Its secretion is strongly stimulated by the presence of partially digested proteins and fats in the small intestine. So as I've told you, pepsin from the stomach is very important because we need to partially digest those proteins. As chyme flutes into the small intestine, cholecystokinin is released into the blood and binds to receptors on pancreatic acinar cells. You see, it goes through blood. It orders those pancreatic acinar cells to secrete large quantities of digestive enzymes, as well as If you recall, this CCK goes to stomach and plays as an inhibitor. Then we have secretin, which has the same pathway, almost the same pathway as cholecystokinin, but secretin is secreted in response to acid in the duodenum, whereas cholecystokinin is secreted in response to partially digested proteins and fats. Secretin in response to acid in the duodenum, so its effect on the pancreas is to stimulate duct cells to secrete water and bicarbonate in order to neutralize our chyme. Please recall that duct cells, the epithelial lining of duct cells, produce those bicarbonates. If you go to gastrin, gastrin is actually very similar to cholecystokinin, and it is secreted in large amounts by the stomach, as we've already talked about it, and it is secreted in response to gastric distension and irritation by the if the gastric wall stretches, and in addition to stimulating acid secretion by the parietal cells, gastrin actually stimulates pancreatic acinar cells as well to secrete digestive enzymes. That's why we mention it here. So this is all about the regulation of pancreas, and I guess this is all about this lecture. Hopefully to see you soon in the next lecture. Bye-bye.